Hey everybody, Mr. Grove here. Today we're going to talk about the cell cycle and mitosis. And the cell cycle is how you make a copy of a cell. And you are very, very good at this. Your body has been doing this since the day you were conceived as one cell, a fertilized egg. And now you have trillions and trillions and trillions of cells as you've made this cycle many, many times. So let's go ahead and start. The first thing you'll want to make sure you know is the cell cycle is usually denoted by a pie graph. And so you want to make sure you can label the pie graph. So we have our G1 phase, our S phase, and our G2 phase, and then mitosis and cytokinesis. And these three phases, the G1, S, and G2, they all belong to a category called interphase. And so this is the period where the cell is prepping to be able to make its copy. And so G1 is basically where the cell is going to stock up the parts and the proteins to be able to make the copy of the DNA. And then in the S phase, that is where our DNA is actually being copied or synthesized. And so you have 46 molecules of DNA since you're a human, 23 from mom, 23 from dad, and you have to make an exact duplicate copy of every single one of those. And so prepping for that takes quite a while, especially depending on what kind of cell you are. After your DNA has been copied, the next phase is the G2, the second growth phase. And so now we have to get ready for the cell to be able to have all of the things it needs to be functional. So like a new mitochondria, a new rough ER, a new set of ribosomes, a new Golgi apparatus. And so the G2 is preparing for all of that. And then mitosis and cytokinesis is going to be the actual splitting part. And so this is where the cell will split, the nucleus will split, and the chromosomes are divided, and then cytokinesis, the actual cell, will split in two. And so that is the cell cycle. Your cells do this very well, but sometimes they mess up and they do it uncontrollably well. And so to try and stop that, we have these checkpoints because we really don't want the cell just dividing uncontrollably around and around and around because that's basically what cancer is. And so these checkpoints will kind of be a stop sign that the cell can hopefully hold that cancer cell in check. So the first checkpoint is the G1 checkpoint. And so this is kind of going up into the end of the G1 stage. And so if we're getting ready to make the copy of the DNA, we've got to make sure we've got all the necessary parts. It's kind of like if you're going to buy a house. So this interphase is kind of like the house buying process. And then mitosis and cytokinesis is kind of like move-in day. It's going to take a lot longer to acquire all the resources and wealth to buy the house. But actual move-in day is usually just a few hours. And so as you think you've got enough money saved up to buy your house, you have that first checkpoint where you go to talk to your banker to see, did I have enough money? And so if you do, you can proceed on and contact a real estate agent and start looking at houses. But if not, then you have to go back and get more resources. So that first G1 checkpoint, once we pass that, the cell makes its copy of its DNA. It starts to prep for all the other organelles that it will need in the new house, so to speak. And then we have the G2 checkpoint. And in this one, we're just double checking, do we have everything for the new cell? Do we have the Golgi? Do we have mitochondria? Do we have all the organelles and all of the DNA copied? If so, we'll proceed to move-in day in mitosis. And so with this, we'll go ahead and divide the cell's nucleus, divide the cell through cytokinesis. And then when it's all said and done, we have the M checkpoint. And with that, we can see, did we do everything right? If so, then the cell's ready to start acquiring more resources for a new cell. And if not, then that cell will probably be terminated or non-viable. So these are the checkpoints that help kind of control the cell cycle growth. Depending on what kind of cell you are, depends on how often you will divide. So for example, a skin cell might go every 2 weeks. A red blood cell might be every 4 months, and that's why you have to wait till you can donate blood again, about 3 or 4 months. A liver cell is actually pretty good at reproducing for organ tissue, so 300 to 500 days, and that's why liver transplants can be cut up and used for multiple recipients. Some muscles, though, are 16 years and are very slow at reproducing, and then some cells, every time they get to this checkpoint, they get diverted back to this G0 stage to where they don't ever go through the cell cycle or divide. And that would be like your central nervous system, spinal cord, brain cells, and things like that. And so that's a way of controlling the cells. Now, when the cell, for whatever reason, and we really don't understand why, starts busting through these checkpoints, that is what's called cancer. And so that uncontrolled cell division will have a lump of cells, a tumor, and if that tumor is malignant, then that can create serious problems for the host organism. And so we'll talk about cancer more on an upcoming video. Some terminology to kind of know. It can be a little confusing because oftentimes we use the same terms to say the same thing, or we use different terms to say the same thing. So we might say, oh, she looks really a lot like her mom. It's just, in her DNA. Or we might say, boy, you can really tell by her genes that she looks a lot like her mom. Or maybe we'll say, boy, she looks a lot like her mom. It's a chromosome thing. We've used different ways to say the same thing, and sometimes that can be a little bit confusing. So let's see if we can kind of differentiate here. So DNA, you had 46 molecules in your cell, and these are going to be long, kind of thin molecules. So we can't visibly see these really with a microscope. But as the cell starts to prepare for cell division and mitosis, the DNA will start to wind around these little spools that are proteins called histones. And so as they wind around, you'll see that they start to become a little bit more visible, the DNA does, because it's thicker now and it's coiled up more. And so as it coils up, that's what's called a chromatin fiber. We'll keep coiling until it gets really, really dark to where we can visibly see it under a microscope. And that would be a chromatid. And in the S phase, when we make a copy of that chromosome, then we would have a second chromatid, or sometimes we call those sister chromatids, and those are connected at the centromere. So that point of connection is the centromere, and that's where the two chromatids, the two copies of the DNA, are connected together. The leg of the chromosome is going to be what's called a telomere. And so that's on this would be, that's where the genes are located. So there's a gene, there'd be a gene, there'd be a gene, and there'd be thousands of genes on these telomeres then. And then the entire thing would be a chromosome. And so that's very visible under a microscope, and this usually has kind of an X-like formation. So hopefully that will help with the terminology some. Now we're ready for mitosis, and it's move-in day. We've got to start to divide the cell. We'll start by dividing the chromosomes that we've copied. And so there are 4 stages to mitosis: prophase, metaphase, anaphase, and telophase. And so we really want to make sure we know that in order. That would be a question you would definitely see on a test or a quiz. So what's going on in this? Well, in prophase, first thing, we have to coil up our chromosomes. So our chromosomes are going to become visible under a microscope. And this nucleus is going to need to start to kind of dissolve away because we're going to have to move some of these chromosomes to each side of the cell. The other thing is the centriole pair is going to form what's called a mitotic spindle. If you remember, the centriole pair was something that showed up back in the old organelle days. And so these mitotic spindles are kind of like fishing lines. And so they got a little hook on the end that will allow them to connect at the centromere and help move the chromosome around the cell. And so all of that is taking place in prophase. Metaphase has a lot of M's, so meta, and you can kind of remember this is where the chromosomes meet in the middle. And so we form a straight single file line of all 46 of our chromosomes. And so those spindles are connected to the chromosome, and that's what helps move them around to get them in a straight line. And the chromosomes are just lined up. Sometimes we call that line the metaphase plate. Next is the A in our PMAT, and that's anaphase. And so you can kind of remember A for anaphase, A for apart. And so this is where we start to pull the chromosomes apart. They were kind of stapled together at that centromere, and now we're going to unconnect them and start pulling them apart. So the mitotic spindle will start to kind of reel in like a fishing line, and we'll pull them apart. Also, we will start to make the cell a little bit bigger. So it's going to start to elongate as it starts to form 2 cells worth. And then our last stage is telophase, T for telophase, T for 2. This is where we're going to start to make 2 cells. And so the 2 cells then, since we're on our way, the nucleus can start to reform. We're going to reel up all the mitotic spindles back into the centriole pair, and the chromosomes are going to start to unwind back into the DNA. And so that is our PMAT, our stages of mitosis. And all of these are visible under a microscope, a relatively simple microscope. So in prophase, we can start to see the chromosomes. Our nucleus is still kind of intact, though. Then in metaphase, we've got our straight line of chromosomes at that metaphase plate where they met in the middle. And in anaphase, we're starting to pull them apart. And then in telophase, we have 2 cells that are starting to form as the chromosomes will unwind into the DNA and kind of get back to normal. But the last thing we have to do is to completely separate the cell. And so that's what's called cytokinesis. And so on cytokinesis, it's a little different depending if you're an animal cell or a plant cell. If you're an animal cell, the cytokinesis will simply just pinch these 2 cells off and make 2 cells out of them. It's kind of like if you have a piece of Play-Doh and you pinch it off enough, it will form 2 cells. What is actually happening is a microtubule is allowed to just kind of cinch off like a drawstring and just pulls it into 2 cells. Plant cells, however, do it a little differently, and they build a cell wall. So it's like we have one giant room, but then they hire some construction and they put a little wall in the middle, and so now we have 2 smaller rooms. And so the cell wall then gets built in between, and that's called the cell plate. And that's how plants go through cytokinesis. So both ways we're going from 1 cell to 2 cells and officially separating those 2 cells where they are now independent of each other. So that is the cell cycle, and that is the process of mitosis and cytokinesis, and I hope that was helpful.